Welcome back to Discovering Academia. We are two college students who travel the world talking with academics about their research, passions, and current events. Today, we talk with Ying Lu, a professor of systems biology at Harvard Medical School.
His work combines physics, biochemistry, and quantitative methods to understand the fundamental mechanisms of biological processes, particularly protein degradation. In this episode, we discuss why curiosity is the most powerful engine for scientific breakthroughs, from the discovery of CRISPR to modern weight loss drugs. We explore the cell's sophisticated quality control system, where a barcode tag called ubiquitin marks unwanted proteins for destruction by a nanoscale shredder. Professor Lu explains how failures in the system are linked to diseases like Alzheimer's and cancer, and how his research could lead to new therapies that either inhibit or boost this process to restore health. We hope you enjoy.
Welcome, Professor Ying Lu. Thank you for coming on today.
Thank you. Thank you for this opportunity. And also thank you for organizing this wonderful webinar series. It's terrific to bring people in academia and to a more audience. Thank you very much.
We'd love to start off by hearing why has it been so important that curiosity be the main driver behind all of your research?
Yeah, so this is a wonderful question. I think throughout my career, most of my work, most of my research has been curiosity driven. And so there are many important reasons. So I would like to just start from some recent examples, for example, CRISPR and also this popular drug, GLP-1 agonist. In CRISPR, we all know that was originated from the study of bacterial immune system, right, or anti-phage immune system. That did not appear to have a strong connection with the applications, but it turned out to completely revolutionize the way people do research, and then in gene therapy, in cell therapy, et cetera, such a big impact. And I mean, the GLP-1 drug, the development of that drug actually originate from the study of a lizard, how that lizard actually would withstand extreme drought and famine. So that doesn't seem that, I mean, back to that days, it's not seem to be strongly connected with applications, but it turns out to be another amazing sort of thing that come to be. So in summary, I dare to say that the most influential, impactful findings in my field is originate from the curiosity-driven research. So I think this is one important reason to summarize is I think the curiosity-driven research likes to explore unknown territories. Just simply the reasons is, for example, if we imagine a well-trained person, if he find a question which is completely unanswerable, curious, right? So that usually means that this is a sort of uncharted or unexplored territories in people's knowledge, and then sometimes that's also a gateway to the entire new world, like the world of leading people to Narnia, right? So this is completely a different thing. So that I would say the curiosity-driven research sometimes can lead to breakthrough discoveries. I think that's actually have a strong impact and benefits. So another also important reasons for that is probably usually typically the motivation for doing curiosity research actually lasts longer, because science is hard and experiment fails, the funding could be rejected, and then so on and so forth. There are a lot of frustrations. So, but the curiosity, people's curiosity, tend to last longer than that. So that actually could be quite important to help people to for them to get enough motivations to even to overcome those transitory difficulties, and then to lead to them to let's say to true understanding, or to eventual getting the problem solved.
Was that curiosity something you always had, or was that something you developed when you were doing your studies?
Yes, that's also, I think, I think curiosity is a fundamental human nature, right? Everybody has curiosity, even our kids, they are very curious, and then they want to know how things works, and then how things happens, and then they are intrinsically curious about. I would say that curiosity is a basic human nature. But also as we grow up, and then there are so many other things that we have to care about, and sometimes the curiosity was not placed as the top priority in our work. And so I mean, one advice that I would have to young people is probably try to not to let this curiosity goes away, right? So try to intentionally foster or cultivate this curiosity. I think they will benefit the people either in academia or outside academia in one way or the other. So this is really a valuable characters that nature give us.
Could you walk us a little bit through your background from physics and then eventually ending up in like systems biology?
Yes, so it's a long story. So I was originally trained as a physicist, and then I studied condensed matter physics, and then at that time I have a limited limited exposure to biology, but I'm very curious because I have a family tradition. So both of my parents work on healthcare, and then I get used to that environment and the knowledge since I was a early age. Then I did my PhD in biophysics program at the Rockefeller University. And then I started to get more exposure to biology or more systematic education and training to biology. But I'm still thinking about how can I possibly combine physics and biology or apply what I learned in the past into solving some challenging biological questions. And so to deepen my education and training, so I moved on to Harvard Medical School and to study biochemistry and molecular biology. And then I stay at Harvard Medical School in the Department of System Biology as a assistant professor, and then to continue my research. And so so there have been gradual transitions of my of my work, going from almost entirely physics modeling driven to somewhere in between, half and half, and then now is more sort of focusing on solving biological questions, but using physical methods and tools, and then we also create new methods and then new tools based on my quantitative background, and then try to apply that to solve the most important biological questions.
Could you explain kind of the role that fundamental science has in our broader understanding of science, and kind of with that, the important role that understanding mechanisms plays in biological processes?
Yeah, I think that's a very good question. And then I can explain that. I can say explain that from using different ways or from different angles. I think one thing that our department of system biology that really focus on is to try to identify the things we call mechanism. Mechanism were called different. You can also call that deep understanding of how things happens. So this is very important in almost any subjects or almost any research areas, and I think in particular for biology, you're typically a biological discoveries that begins with a correlation. For example, we find there's some diseases that correlates with for example, the change of temperature, or for example, the change of people's diet, so on and so forth. So this is a correlation, right? So we don't know whether this is really there's a causality between, or this is just simply correlation, right? Maybe in many cases it's simply correlation. So, but in order to transition that into a therapy, and then try to understand whether this X actually leads to Y, and so we need a mechanism. So we need to know how things works, and then for example, why the changes to people's diet, eating behaviors would somehow cause a certain type of disease, whether there's any causality. So we have to understand. So what part of our human body, if goes wrong, will cause that type of disease, like diabetes, obesity, so on and so forth. And so this is called the mechanism. So essentially, it's seeing how it's basically interpretation, fundamental interpretation of how things works or how things could happen. And so this is an important part of our research and try to understand how things works. So in particular, in my own lab, is to understand how the protein degradation system works.
Yeah. And then do you think the focus on mechanism can ever go too far where researchers get siloed into a certain way of thinking and they lose track of the larger picture?
Yes, that's also a question, or I think issue that many people brought up. Yes, I think you are right that if this study of the mechanism, probably there's never going to be an end, right? There could be mechanism, and then there could be mechanism of mechanism. There could be mechanism of mechanism of mechanism. So the things could be very deep, and then could be very detailed. And if a researcher, a magnetic researcher like myself, want to go into that route, so I can spend decades on a single question, become deeper and deeper and deeper, right? And then, as you said, unfortunately, a sad effect of that will be losing the big picture. Right? I wouldn't know how my research would be able to translate. It would be able to benefit the society. Although I think it does probably solve the fundamental questions, but it gets a penalty of that is get detached from the society, which is not good. I think for the well-being of one research lab, and also not for good for the well-being of the entire research society as well. So that's why I think that may connect to your following questions that I think having experience, having a knowledge in life science industries actually help tremendously in connecting my own research and connecting my mindset, lines of thinking with how our research could better benefit society, could translate. So why people are being thinking into very tiny details, to just keep the, as you said, keep the big picture in mind.
Do you think enough like fundamental researchers keep their like potential translations in the back of their mind? Do you think like that area is too focused, broadly speaking, or do you think they do a decent job of being like, this is why I need to understand this mechanism for like these broader applications?
Right. So my experience is this. So in this environment where I was trained, I would say since my undergrad and my PhD, almost everyone around me, they're all very great scientists, and so their research is, I would say, during that period, the academia and industry is somewhat independent. So they are moving with their own pace. So they don't really have a lot of overlap, at least within the environment that I got trained in. And then usually the people around me, those great scientists, when they try to ask questions, and then they try to define their research areas, they tend not to think too much about whether this is going to be important, whether this is going to translate into important therapy. But you know what? Very often these things can be translated eventually into into important therapy or some kind of medical device. But this may not be done by this lab per se, or maybe done by some other labs that that happen to read their publications and then find, oh, this is so interesting to me. I would continue working on that. But the people around me are mostly focusing on the basic questions, as we call them curiosity-driven research. And so they would search around, and then they find whether this is there is important unknown territories, as there has been uncharted research areas that has not been explored before. And then let's just go into it and then see what we can find. And then, interestingly, this seems how science works. And very often, this seemingly trivial and then just completely curiosity-driven studies actually can be translated into something very important.
Then diving into your work specifically, you mentioned protein degradation. Could you compare and contrast protein degradation and synthesis?
Oh, yes. Protein degradation and synthesis is like the yin and yang of like the two sides of the same coin, I would say that. Because in order to precisely control the protein levels, right? So there need both the precise control of the synthesis rate and also the precise control of the degradation rates. And I think the synthesis system and protein degradation system are also strongly coupled, and then they talk to each other, and they are both intimately connected with human diseases. So there was estimation roughly 40 or 45% of human diseases due to failure or malfunctioning of the degradation system, those machineries. So, which is not surprising because those quality control and then degradation systems is indeed play quite important roles in almost all aspects of cell biology, human biology.
So how does, just for like maybe the audience that might not fully understand biology, like what do the processes look like at a high level?
Yes. So how this degradation works is typically what we talk about is we call them regulatory protein degradation or precise or precision degradations, simply speaking. And so we are not talking about this protein degradations, which is non-selective. For example, under stress conditions, the cells may undergo autophagy process that degrade a whole chunk of cells. Also, it's non-selective. So, but under the normal conditions, the proteins needs to their levels or their stability needs to be regulated. So usually the beginning of this degradation process is the cell would recognize what things needs to be removed, and then there's a demand. So and then how the cells tell the degradation system which protein to be removed is by putting a tag. This tag is called ubiquitin. Ubiquitin is a small proteins, and then so this ubiquitin gets covalently coupled to the target proteins, but this ubiquitins can form a chain, and then those there are usually multiple chains. The chains could have different topology, so they have a variety of different forms, a large number of different forms. So this some people have called that ubiquitin code. So you can see this as a barcode that the cells put on the protein target that needs to be removed. So this barcode contains the information, tells the degradation system how this protein needs to be degraded, and then when it needs to be degraded, at what rate it needs to be degraded. So presumably all the informations are there. It's just that the degradation system, for example, the key degradation machinery is the proteasome, which is a nanoscale. You can see that it's a nanoscale machine or nanoscale robot in the cell that consumes energy. And then they recognize this barcode that's put on the protein targets. And then every second, there are hundreds of millions of protein molecules in each cell that need to be removed or recycled by this machine. So it's a large number of different molecules need to be done. And then they all go through this proteasome system. And then this nanoscale machine will recognize that barcode and then engage these protein targets, unfold them, translocate them into the central chamber, just like a shredder, and then cut them into pieces. And then those pieces get released, right, from the proteasome eventually, and then that gets recycled back into amino acids. So this is the brief overview of how the degradation actually works.
And you mentioned that there's different I guess, like, ways that it gets degraded, and you said, removal and recycling are at the end of the day, are the amino acids all end up being recycled, or do some of them just get removed fully from the system? Or what do we know about, like, those distinctions so far?
Yeah, that's actually a very interesting question. It's also active research areas, and so there are multiple ways these things could work, and I think by in the ballpark. So most of those proteins that are degraded, and then their amino acids get recycled back into the system, and then to build more materials. And then, but there's a small fraction of those peptides that's cleaved by the proteasome from those target proteins play a very important role. So what they do is they get presented to the immune systems, right? So we all know this T cell receptor, how those things works, and then those T cell receptors, and then the MHC complexes on the cell surface, and then almost every cell, and then those MHC complexes will present pieces of the proteins inside the cell and tell the immune system, okay, this is am I in the normal state or in the abnormal state, and the immune system will deal with that dysfunctional cells. So this is how those things work. Has been simulated, work very well. And then so that piece of peptide presented by the MHC complex are actually mostly from the proteasomal degradation, and then a tiny fraction of those peptides gets translocated to ER, and then they bind to MHC and then presented to the cell surface. So without the proteasomal degradation, there's essentially you can say there's no immune presentation.
So on that, if we think about ubiquitin tagging internal like organelles for degradation, some of those going out to be presented to the T cells, would that be how the larger cell as a whole would get degraded if it needs to?
Oh, to some extent, to some extent. So this is a part of the immune recognition, and then so this peptides presented degraded by the proteasome and then presented on the cell surface. You can think about this as a barcode of the cell itself, right? For example, if there's cancer cells, cancer cells, there's chaotic states, and then the degradation machinery sometimes behave very differently from the normal cells. There's also creations of cancerous fusion proteins that does not exist in normal cells. So those created, we call them neoantigens, which means antigens that's not there in the normal cell, but in the cancer cells. And then on the cancer cell surface, so those can be recognized sometimes by the T cells, and then if they recognize as a new antigen, they would kill the cells. So that's actually an important mechanism, part of the mechanism as our natural defense mechanism to prevent cancer from occurring.
You mentioned that 45% of the diseases we know are related in some fashion to degradation. I know that maybe isn't specific to protein degradation.
Yeah.
But within protein degradation, do we know, like, if there is a failure in the degradation system, how much of that is coming from ubiquitin versus proteasome?
Right. How much of that was from ubiquitin or proteasome? So this could be I think the answer is hard to give a general answer. But usually there are two main degradation machineries. One is the proteasome, and then the other is autophagy degradation system. So both degradation systems are controlled by ubiquitination. Although there's some ubiquitin code, barcode leads to proteasome degradation, leads to autophagy degradation, but they all controlled by ubiquitin. They're both controlled by ubiquitin, and also as well there are diseases, human genetic disorders, for example, that mutations cause, which is more associated with autophagy system, and then they are also more associated with proteasome system. So we can find both examples, and then they cover a broad spectrum of human disorders like neurodegenerative diseases, even neurodevelopmental disorders, type 2 diabetes, for example. So there's a variety of different human disorders that people can find the root in some problem with the degradation machineries.
So is autophagy always the entire cell, whereas like proteasome would just be like specific targets within the cell?
Not always. So there are
Okay.
several types of autophagy, and then so there's the one traditionally known autophagy called macroautophagy. So that usually means the removal of a chunk of the cytosolic uh, um, content without much selectivity. So even that macroautophagy, there's some they're not entirely non-selective. So there are some evidence they may be partially selective, partially non-selective. But there are also branches of autophagy that are very specific, very selective. For example, ribophagy, or ribophagy means the removal of ribosome, defective ribosome by autophagy, or mitophagy, which means the removal of defective mitochondria by mitophagy. So those kind of by autophagy. So this process are controlled by ubiquitination, and then they are very specific. They only remove the defective content. As said, so autophagy can be selective, and which probably not surprising. Most of the selective autophagy also controlled by ubiquitination.
And then if we're thinking about proteasomes breaking down the protein similar to like a ribosome building a protein, is
Right.
there an analogous structure in autophagy that's breaking down the targets?
Autophagy works a little bit differently because autophagy, that's actually an interesting question. So, the proteasome and ribosome, the protein creation, and there's actually quite a lot of similarities. The structures are very different, but if we're talking about the physical principles behind this, or we call that designing logic behind this machinery, there's some similarities. But autophagy, interestingly, seems to work by different rules. And I think it has been less studied, but usually autophagy is the initiation is a high density of ubiquitin or ubiquitin chains on the targets, and usually those targets are hard to be removed. They're big, and they're hard to be removed by the proteasome, and then that triggers a series of downstream reactions events that eventually cause that a target to enter the endosome and lysosome, and then they are degraded by the protease inside lysosome. And so this is typically how the autophagy works. But proteasome and protein synthesis are controlled by protein machines. So they are the machineries, they are the nanorobots that in the cells that control this process. Certainly, the question is why the nature actually design or actually evolves those kind of machine-like entities to carry out these important tasks. So that's I think a deeper question. So there are many answers to that. But I think one is those machine-like entities, in contrast to, for example, 10 or 20 separate enzymes carrying out the same reactions, why the things doesn't work that way. So this assembly line, like protein machines, I think they are easier to achieve. We call them control precision. So they are good for ensure this process happens precisely, because like a protein synthesis or protein translation or transcription, so there is a proofreading steps. So if things go wrong, and then the ribosome and then polymerase could backtrack and then correct the mistake. And then there's also similar steps at the proteasome. So I think they all probably engineer has been evolved to make sure that this important process actually happens reliably.
Do we know what role genetics or epigenetics play in protein degradation?
Yeah. Yes. This is I'm not aware of any direct role of genetics. Certainly, genetics and epigenetics, everything, and then they cause the synthesis or creation of the machineries, proteins involved in protein degradation. All those things we mentioned, ubiquitins and then the proteasome, ribosome, they come from the genes that needs to be translated to proteins, and then so in that respects, and this epigenetic and genetic regulations are very important. So eventually, this whole thing is a feedback, right? So, and then yeah.
And then, are there environmental factors like fasting per se that cause more ubiquitization or cause more protein degradation than other things, or like maybe heat, that type of thing?
Maybe heat. Yeah. This is right. This is a little bit outside my field. I think so. Mostly what I read or what I know is so I have not directly looked into the effect of nutrient or heat in this degradation system, but I think the short answer is definitely. So the nutrient and then fasting, nutrient levels and fasting, and then the stress conditions play a very important role in controlling. There's multiple pathways like mTOR pathways, like AMPK signaling cascades that are very important in controlling the rate of protein turnover, so which could be both protein synthesis and protein degradation, and then to help the cell to maintain a homeostasis. And then, but you can ask how the proteasome system actually play a role, and in response to nutrients levels, starvations. I think this is actually an excellent question. It's actually an active research area at this time.
Interesting. The reason I asked that was I know in like the health space, a lot of people talk about fasting causing like autophagy after a few days.
Right.
And that was kind of the impetus on that. I didn't know if we understood any of the mechanisms beyond just we can see that there's less cells. Like,
there's some understanding, but I think there is still there are a lot of important basic questions that remains. Probably not surprisingly, the cells has evolved a very complicated machineries to sense the nutrient levels. The cells require so many different nutrients, right? Acid levels, sugar level, nutrient level, ATP levels. So they all have a defect sensors. Each have different sensors, and then but all those informations and then need to be integrated, right? The cells like computer, they have to integrate all those informations and then make a decision what to do, right? Do I need to keep dividing, or do I need to enter different nascent states or die or whatever? So all those decisions are made, and how those decisions are detected, how the cell actually cope with the change of nutrient levels, I think is very interesting, important questions, but need more study.
and then kind of on that, I know you guys have made a simulation of the ubiquitin like proteasome interaction and degradation of proteins. Has there been a simulation of an entire cell like completely functioning?
Yeah, there have been some studies like that. There is a study, for example, there is several years ago, there is a study, I think at Stanford, called virtual cells or in silico cells, something like that. So people, as you said, they aim to simulate every pathways that available in a bacteria or eukaryotic cells, and then they put them together. So there are hundreds, if not thousands, of pathways, and then put those pathways together, and then to see whether this actually could recreate, from bottom up, and to regenerate the behaviors of bacteria or cell that normally behave. I think there are some success of that. Yes, I think it's a very interesting area. So if we can simulate, if we can predict how a complicated biological system actually works, that will be quite important, quite beneficial for both research and also for industrial applications.
Too soon with AI.
Yeah. Yes.
Yeah. We talked about the role of translational research earlier. What are some of the ways you imagine your work might translate to human health?
Yes, I can imagine. This is also an important question that we are thinking about right now. Fundamentally, there are many ways that our system could, our studies could be directly or indirectly translated into human health. So one example is, one research we have been doing previously is to study how the proteome works, and one particular question we have been thinking about, and also other people have been thinking about, is, for example, there are hundreds of different human genetic disorders can be mapped to proteome mutations, point mutations on the proteome. So certainly there's a cancer aspect, but even beyond that, and then those mutations map to different subunits on the proteome. And a lot of those are neurodegenerative disorders or autoimmune diseases, so on and so forth. So in most cases, we have no idea how those single amino acid changes could lead to such dramatic detrimental effects. So this is, and then we try to understand, okay, if we better understand how the proteome machine works, and then maybe we have a way to understand the disease mechanisms, and then find a way to actually to treat those diseases. And the other way, for example, we have been thinking about is one area that we have been currently actually working on is this concept we call that difficult substrates. And then because we realize that the cells contains targets of the proteasome that are degraded by the proteasome, but not by the proteasome alone, needs some help. So unlike perhaps most targets that the proteasome can handle it, but those difficult substrates, some of those are membrane proteins, some of those are protein aggregates, some of those are protein complexes. So, and the proteasome can degrade them, but it requires assistance factors. And interestingly, many of those difficult substrates has a connection with diseases. One example is the protein aggregates that occur during aging and then during neurodegenerative diseases. So proteasome is an important factor that keeps those misfolded toxicity proteins in check. But that process is not 100% reliable as people get old, and then the proteasome activity decreases. So which may partially contribute to the accumulation of those aggregated proteins. And then we have been interested in what the cell has been evolved, the system that helps the proteasome to tackle, to degrade, or to disaggregate those protein aggregates, toxic protein aggregates. And then, so once we identify those factors, and then we can find a way to see how can we boost the activity of those difficult substrate degradation machineries, and then so which may be able to help with certain conditions that is associated with the accumulation of those toxic proteins.
Would one of those be like the amyloid beta plaques for Alzheimer's?
Yeah, amyloid beta is one example. Although amyloid beta, most people would think this is mostly occurring outside cells.
Yeah.
But there is a counterpart of amyloid beta is called tau protein, and then that is mostly in the inside neurons. And then, yes, you're right. So proteasome is a major factor that degrades tau protein aggregates that contributed to a variety of different neurodegenerative diseases like Alzheimer disease and frontotemporal dementia, so on and so forth. And then this is actually one of the direct areas that we are studying right now. So we want to see whether boosting this difficult substrate degradation system could help the cell to get rid of tau protein aggregates. So let's see what happens.
Yeah. Because is the idea that the tau protein aggregates then become like amyloid beta plaques like outside the cell?
Tau and A beta are intimately connected. Tau dysfunctions could lead to changes of A beta processing and expressions, and also vice versa. So they could also seed. They could cross seed the aggregation of either one. But the exact mechanism that whether this is tau or A beta is still active. I think is still active research areas in Alzheimer's disease field. So I think it fundamentally there's very strong evidence that the tau and A beta aggregates occur simultaneously. There's a high degree of correlation of those two aggregates. It's just that A beta is mostly outside the nucleus, outside the neurons, outside the cells, some inside the cells. But tau is mostly inside the neurons.
Okay.
Right.
Yeah. And then you've also talked about the ubiquitin proteasome system being a popular drug target. Could you explain like when you'd want it to be an inhibitor versus like an activator?
What activator? Yeah. Right. I can give you examples of, one example is cancer that, for example, we have talked about. Cancer you can think about as a ruling society. There is no regulation, right? The production and recycling basically happens in sort of chaotic, random fashions. So certainly this society can be invasive, can be really cruel from outside because there's nothing can control them. They don't listen to anything, and then but they're also fragile inside because those things that maintains the well-being or the mere existence of that society is sort of in a kind of disordered fashion. Both the protein degradation and protein synthesis machineries. So this we call them vulnerabilities of cancer cells. And as one can expect that the proteasome is one of those vulnerabilities of cancer cells. For example, in melanoma cells, and then in normal cells, the proteasome or the protein degradation system has some capacities. It's not entirely occupied or overloaded, so that it has some capacity to deal with unforeseen conditions like stress conditions, things like that. But in the cancer cells, this degradation machineries can be just overloaded. And then so that makes them sort of more vulnerable to compared to normal cells to proteasome inhibition. And then as expected, one of these great applications of this proteasome inhibitor that was pioneered by Dr. Fred Goldberger at Harvard is to treat myeloma and lymphoma cells. So this because those cancer cells are very sensitive to proteasome inhibition. So a slight inhibition, normally the normal cells can tolerate, but those tumor cells will undergo apoptosis, will die. And so this is one example of using how to explore this selective vulnerabilities in cancer cells by targeting the degradation machineries could lead to important therapeutic outcomes. I think on the other hand, there are conditions associated like aging, neurodegenerations, and also some type of sometimes it could be diabetes that is caused directly or indirectly by a failure or the reduction of the activity of the degradation system. So in that case, and then when we think about, okay, can we actually boost the activity of the proteasome or autophagy machineries, for example, using the PROTAC molecular glue, this is one example. But there are also examples of drugs that can activate the proteasome globally. So that has been shown, at least in animal models, that could ameliorate the phenotype of Alzheimer's disease by increasing the activity of the proteasome. So there are many examples like that. I think I think summary is this degradation system activity must be carefully controlled. It cannot be too high, it also cannot be too low. And but there are also pharmaceutical interventions we can implement to tune the activities, and then so that we can use those to treat different conditions.
Are any of those pharmaceutical applications developed or close to development yet?
Sorry. Any of those? Yes. The proteasome inhibitors, for example, bortezomib has been FDA approved for a long time, and they have been used to treat a variety of different cancers. And there are also other drugs like cafuzumab, which is different versions of proteasome inhibitor. Moretumab, which is a brain penetrable proteasome inhibitor, that has been under clinical studies right now. And for proteasome activators, there are I think it has been mostly in the research stage, so that has not been approved, any drug approved. But in the animal models, I think from the early evidence, and I think this is look like promising that there's some way if we can activate the proteasomal degradation, it could help people. It may be able to, I hope, to help to treat certain conditions.
Yeah. And then just taking one step back, I want to clarify the inhibitors. So if we think of like cancer as like an overactive like growth, right?
Right.
Wouldn't we want to degrade more proteins, or like would we want to degrade the entire cell? Like, why is it an inhibitor on that one?
Oh, why in the cancer is an inhibitor? Okay, so I think simply put, I'm not entirely sure about the question you asked, but I think simply put is the cancer cells, they tend to synthesize a lot of proteins that they don't need, and then they need to degrade it, so which results in the degradation system is overloaded. And then, so if we inhibit it, for example, maybe 5 or 10% inhibition, minor inhibition of this degradation system will lead to a detrimental effects, collapse of the entire cells, and then either make them not able to divide, or make them to undergo a programmed cell death. But this minor inhibition somehow can be buffered or sometimes tolerated by the normal cells.
Okay,
this is fun. Yeah.
So if a cancer cell is growing like too fast and the proteasomes are working too hard, then it kind of stays at the level where it can continue to grow and populate. But if you let it, if you inhibit the proteasome, you let it grow too much where then it kind of gets overloaded and then it has to like kind of kill itself.
Right, exactly.
Yeah.
I think we might have already answered this in the discussion, but looking into the future of your research, what are some of the biggest questions you're hoping to answer?
Yeah, there are several interesting research directions we are following up, and then certainly we are still very interested in how the proteasome works, and we want to understand how this nanoscale protein machines could recognize the barcode of ubiquitins on the substrates and then precisely execute their job of protein degradation. So this is sort of deeply mechanistic questions that we try to pursue. And then, but we are also interested in this thing called difficult substrates, or this machineries, either factors or additional steps in the cells that help this proteasome to be able to handle other challenging targets but not be able to degrade otherwise. And one of the reasons is that this mechanism has been poorly understood. And the second is many of those, we call them difficult subjects, has close connection with human diseases. I think hopefully that through these studies, and I think our research will be able to find a room to benefit the society.
I think that gives us a pretty good overview of a lot of your work. And I know we wanted to touch on one other thing is in the last few years, you started to work as a scientific advisor with a private equity firm.
Right.
What is it like being a scientific advisor? How did you get pulled into that? And like, because I think that's a role that not a lot of academics always talk about.
Right. Yeah, I can give some thoughts, and because I was pulled into character T by a friend. So initially, she was asked me to give some advice about the operations, mostly scientific advice about the operations of the investment firm, and then to help them to answer some scientific questions. And then after a trial, I find this is quite interesting. So I would perhaps want to get more engagement with operation of the firm. So they sort of decide to stay, and then to establish this scientific advisory role inside the firm. and I think is so far has been quite interesting. It's a mutually beneficial process. I think on one hand they can get my opinions, and but often the topics are quite diverse. So very often it's outside my expertise. So in that case, I could refer them to people that has this knowledge, could help them to answer questions. But also for me, this is also a quite important learning experience, because I think on one hand, they help me to sort of motivate me to study, to learn the areas that I may not able to have to do that without this kind of experience. And I think on the other hand, it sort of motivate me to think and what our society really need, and then how I could somehow translate my basic research into something that could impact, that could be benefit the society. So this, I think, is eventually it's a mutually beneficial process.
Yeah. And earlier in the conversation, you mentioned that the traditional model with academia is that academia and industry were independent. Could you shed some light into kind of your interpretation of the modern context now and the relationship that academia and industry have with each other?
Yes, I think there has been a lot of changes since I was trained, since my training stage, and there are still a lot of, there are perhaps even more bigger changes going on at this time. And I think in general, I can see the trend that the border between academia and industry become blurry. So there's more communications, there's more interactions between research and academia. It's happening right now, and then I think this is probably going to be the trend, and I think this is also going to be beneficial for probably for both parties, because the industry really just benefit a lot by having open-ended, inquisitive research activities, and then help them to navigate their any kind of their specific goals. And then, but I think academia, on the other hand, by collaborating or interacting with industries, would benefit by, for example, receiving a better support or advice from the industry. And then all those things ultimately would translate into a stronger impact of their research, and then the stronger translational potential, stronger impact, or strong applications of the research from academia. And I think this is a good thing to happen, to see. Yeah, I want to see more collaboration or interactions between academia and industry in the future.
And then I think nowadays, in like the era of like fake news or conspiracies, people always get like worried about, oh, the science is going to get manipulated by industry and profit motives. Do you think like that's an overstated like fear? And do you think maybe industry partnering with like fundamental scientists, that fear is there's a lot less likely to be, like have a negative influence? Because if you're trying to understand the like, fundamental mechanisms, that can't be a drug that they're going to profit off of or something like that.
Right. I think the question you brought up is very important. Yeah, I definitely see examples. I read examples of that. I think the industry would somehow influence the academia, the research, or manipulate, manipulate the outcome of the research in some way that would not be trustworthy. And I think those kind of things happens, unfortunately. And I think this kind of things may still happens even without collaboration between industry and academia, because ultimately academia, in order to keep moving forward, they have to be funded somehow, right? By most likely not entirely by the government, and then there have to be some funding agency. So I think those kind of things are hard to avoid. It's not going to, even if we stop the collaboration between academia and industry, it's not going to stop this from happening. So that's, I think, in order to minimize this potential thing, potential downside, and then there has to be more regulations, perhaps, unfortunately. And then, but also, so that's a whole different topic. I hope to touch on if there's opportunity next time. But I do feel like, for academia, there are many, there are countless number of papers published every year, right? So the applications of the research, at least in biology, is one of the ultimate exam of whether that knowledge is called that useful or is called that true, because some of this or perhaps many of these models that we created are not wrong, but it could be incomplete, and or it could be it could be, sometimes it could be misleading. So the question is, how do we know that? There actually, unfortunately, there's not many ways that we could know whether our knowledge about the biological system has already been completed, or there may be still important missing links there, unless we put those knowledge into real applications. So which means we need help from industry to translate our knowledge into drug or into a device, that to put them into the most strict test, which is the clinical studies, right? And then, so if those drug works, that means, okay, yes, I think our knowledge is most likely to be correct, and then it has because it translates to something very useful. But on the other hand, I think that indicate that there may be something important that's missing in our understanding, and then we should work harder to get better knowledge.
I think that's a really good way to conceptualize that relationship. As we wrap up, do you have any advice to the listeners?
Oh, yes, I think there are many things I want to say, but especially for the young fellows, I would say just keep following your original motivations, and then find opportunities to booster or to foster your creativity or inquisitiveness. And then please don't be afraid of working on basic research. So there are a lot of interesting things going on. There are a lot of opportunities. So try to keep going. Try to follow your own instinct. Right? Don't be afraid that this is basic work, so we actually not work on that. I think this is not the right thing to do. So and then we need more people. Then we also need more support, people joining the basic research studies. Even for a short period, even for a few years, it will probably be really beneficial for one's career. If you look at even regardless whether this person is going to industry or the academia in the future, I think have some experience in basic studies is going to be very, very beneficial. Right.
We appreciate you coming on today. Thank you.
Oh, thank you so much for the invitation. Okay. Very, very good conversations. Thank you for the great questions. All right.
Thank you.
Keep in touch.
Bye.